Welcome back to the channel, and this video is about Warburg effect in the oncology series of videos. The next important hallmark which we are going to discuss in this video is the reprogramming of energy metabolism or the altered energy metabolism which is observed in the cancer cells. So before getting into depth of what actually is Warburg effect, let's discuss in some common terms about this Warburg effect. What is Warburg effect? Warburg effect is actually the phenomenon which is observed in the tumor cells that seems to make to think that this tumor cells are addicted towards glucose and towards the glycolysis. Yeah, it's the right word used. It's called addiction because in spite of several other good pathways or several other pathways that are much yield, these tumor cells are highly in need of this sugar molecules or the glucose molecules, and they only carry out their process of breakdown of glucose through glycolysis. This trait of cancer cells are first reported in 1924 by Otto Warburg, and he was the Nobel Prize winner for finding the respiratory enzyme called the cytochrome C oxidase in ETC. Fine, now let's see what happens in a normal cell under adequate supply of oxygen. First, glucose which is present in the blood enters into specific cells through the specific glucose transporters, maybe which is present largely which is GLUT1, and then enters into glycolysis. In glycolysis, it is actually breakdown into two molecules of pyruvate, and this pyruvate molecules is then processed and transported into the mitochondria where it is changed to the acetyl CoA, and this acetyl CoA enters our Krebs cycle. And in Krebs cycle, there is a subsequent release of an carboxyl group as carbon dioxide in each round of the cycle, and a subsequent release of several ATPs, which is known as the energy currency of the cell. So the energy within the glucose bonds is then converted into ATP and can be saved as energy currencies in our cell. But what actually happens in cancer cell is quite different. And excuse me, it's not quite different. It's totally different and totally contrast what happens in a normal condition with the adequate supply of oxygen. Let's see what dramatically and drastically happens wrong. We all know that in normal cells which do not have adequate supply of energy, that is known as something of the hypoxic conditions, the normal cells will channel the pyruvate to the production of lactate, and this lactate is then again rushed back into the blood, and this blood with the lactate is taken to the liver, and the liver again converts this lactate back to the glucose. So this is something known as a Cori cycle, and this makes a subsequent supply of glucose within the body and keeps it as a cycle. And for some reasons, our cancer cells, even though with a subsequent or as a sufficient amount of oxygen, they do follow the same pathway and do not channel pyruvate into TCA or the something known as a Krebs cycle for which yields more amount of ATP. The difference in the ATP production between these two type of cells is totally contrasting. While in the normal cells, where the each glucose through glycolysis and subsequently through the Krebs cycle can yield up to 36 ATP, considering ATC into also an account. Whereas in contrast, if you see in cancer cells, it's only 2 ATP. So why are they compensating such an increased amount of ATP, which could be brought but through channeling them to through channeling them to the Krebs cycle, which may be of very much help in such a very rapidly dividing and proliferating cell. Not only in the process of glycolysis, they are also very hungry towards glucose. And how are they hungry? They express a very large amount of glucose transporters, that is known as GLUTs. And these GLUTs are very much higher in these cancer cells. And radiologists exploit this property of these cancer cells for the identification of, for the spotting of the cancer cells. And how do they do it? Positron emission tomography, that is PET scans, actually work on this basis that the tumor cells exhibit a large uptake of glucose than most of the cells. So what they do is they inject a glucose analog which is known as 18 fluoro 2-deoxyglucose, that is FDG, into the bloodstream, and it is converted by the glycolytic enzyme hexokinase into FDG phosphate. It's converted into FDG phosphate inside our cells after being updated after taken into our cells, and this can be visualized using PET scans. And this phenomenon of Warburg effect finally lead us to three questions which are unresolved. why do like mostly nearly 80% of cancer cells metabolize most of their glucose via glycolysis when completion of glucose degradation in mitochondria by TCA would afford them vastly more ATP to fuel their cell growth and proliferation. Maybe let's consider this to our first question. And second is aerobic glycolysis, that is a Warburg phenomenon required for the maintenance of the cancer cell phenotype, or does it represent nothing more than a side effect of a cell transformation that plays no causal role in cell transformation and tumor growth? So first one is I already discussed. Though there can be more ATP production via Krebs cycle, they don't do so and they still restrict themselves to glycolysis, and that is to inadequate supply of oxygen. That's it's called aerobic glycolysis. And why they do so? That was first question. And second question is such transition of movement from normal cells to Warburg affected cells provide them any edge over normal cells for their maintenance of their cancer phenotype. And third, that it doesn't have any effect, but it's a side effect of being a cancer cell. And now let's see the possible explanation for all these. Fine. Moving on to the first explanation, or the first possible explanation, comes from the observation that the cancer cells within a tumor often does not have some inadequate access to oxygen. So cancer cells would seem to have well adapted to this oxygen starvation by developing Warburg effect. Since glycolysis operates normally under hypoxic conditions, but this explanation still has a defect and raises another question. That is, this explanation still fails to explain why cancer cells, even when provided with abundant oxygen, do not take advantage of this oxygen to generate ATP in far larger quantities. So let's move on to our second explanation and the more simpler and most accepted explanation. And this explanation is derived from the fact that glycolysis actually serves a second role apart from ATP generation, and that is, it provides the intermediates in the glycolytic pathway, which functions as precursors for many molecules which is involved in cell growth and its proliferation and in many biosynthetic pathways. So, by blocking the steps in the glycolysis, by blocking the last step in the glycolysis, all the further steps which are present earlier and its glycolytic intermediates can then be channeled or diverted into many other critically important biosynthetic reactions. And this behavior totally contrasts with normal cells, which generally are not proliferating and they do not require such large set biosynthetic reactions or normal cells, and largely only depend on their ATP to sustain their normal metabolic activities. And by some statistics, normal cells use more than 30% of their imported glucose to make ATP, while cancer cells only use only 1% of their glucose for this purpose of producing ATP, so rest of them for biosynthetic reactions. So you can see the vital role of the distribution of the intermediates of the glucose, or the role of glycolysis in two different types of cells, that is one in the normal cell and one in the cancer cell. And hence we can elicit that the Warburg effect is necessary for the survival of the cancer cell in order, in a way such that the Warburg effect supplies them with enough biosynthetic intermediates for their biosynthetic reactions.